There we go. So this one is called In Vitro Virucidal Assays to evaluate the antiviral efficacy of nitric oxide nasal spray against different SARS-CoV-2 variants. So a lot of people use nasal sprays for preventing COVID. I'm very skeptical of these, so I decided to pull some articles. This one is a preprint. I don't typically like preprints, but there actually wasn't a lot in the literature on these nasal sprays. So when it's slim pickings, you just kind of have to go with what you got. So I'm just gonna read through. I'll highlight stuff while I'm going for when I do my video later on. Cool. So with any paper, they always start with an abstract, give you a little background context, do a materials, results, and conclusion. Then they have keywords. This is just for search terms. This isn't really useful for when you're reading, and sometimes they'll break down abbreviations if they're nice. Typically, you only see that in theses. You don't really see that in the actual papers themselves. That'll probably go away when it goes to a peer-reviewed publication. I wanted to buy Novade, but it's so expensive. Yeah, and I'm still skeptical of the efficacy because we continually produce mucus. I suspect that it would become ineffective very quickly, and that's sort of my question is how long will it actually be effective for? Because I do think these things will be effective for short periods of time, just not long periods of time. And that's my worry is that it'll give people a safe, a false sense of security. Cool. So let's zoom in a little. That looks good. You guys can read that, I think. So going through the background, so it's just talking about the mutations of COVID, that it's rapidly emerged. Nitric oxide nasal sprays have been found effective in reducing the intranasal viral load. So it's specifically looking at people that have COVID and then reducing the viral load. This is not necessarily preventing COVID in people that don't have COVID. So that's a very important point. So I'm just going to highlight that. Use yellow. I have my own personal color coding system. Yellow is just general facts. Green is something I really think is cool. Red is something that's really cool but is not so good information. And then let me just do a little quick save. Because I save it on my desktop, I have to save over the file that I originally started with. I'll just double check comments. Cool. All right, so and it's claiming also expedited viral clearance in clinical trials, so I'll continue that on. So they're looking at in vitro effectiveness in neutralizing Delta and Omicron variants. So this is just after the original strain. This is right after the Wuhan. This is very early on in the pandemic, so this would be around 2021 that they're looking at in terms of the time scale, even though this paper is published it's not published yet, but this is going to be a 2023 paper. So that's just kind of the grounding context you kind of have to think about. So cells were incubated with increasing concentrations of nons. What are nons? We look through that nice little thing. Typically, you don't just introduce a thing. You'll write the word and then you'll put in parentheses so that you don't do that. That's why you only really see this type of thing in theses. So nons is nitric oxide nasal spray. Because then it's just annoying because people are, it's right there too, nons. So whatever. So they were exposed to 10% nons with media and for different durations. So this is basically saying that it's a 10% concentration nasal spray with the nitric oxide, and they're testing at different time periods. Testing for different durations. So basically this would be like spraying your nose we'll have to see. It could mean that it's spraying your nose for 5 times versus 10 times, or it could mean we're testing at 5 minutes, 10 minutes, 15 minutes. We'll have to see what they mean. So subsequently incubated with cells. So they're exposing COVID, likely in some sort of petri dish. So it's an in vitro study, and they're incubating it with the Delta variant and Omicron variant BA1 for 48 hours. That's how they're testing it. And then I'll just see if anyone has any comments. Nope, we're good. So that's what they're doing. And then they're comparing this with a control. Oh, so while the viral copy number and the cycle threshold CT values from the cultures were analyzed to determine antiviral activity of the sprays against Omicron variant, the active comparators. So this is their controls. They're Whoa, that was weird. They're comparing against 70% ethanol as a positive control, and then phosphate buffered saline solution as a negative control. So that's good. They have two controls. That's very nice. You always like to see that. So basically this positive control, ethanol, will definitely kill COVID. So that's going to be showing that, yes, we are actually killing COVID. Phosphate buffer solution, that's just a salt solution, so that shouldn't have any effect on COVID. So that would be it surviving for this 48-hour period without doing any damage to the COVID virus itself, theoretically. So you should have very high levels in the phosphate buffered solution. You should have basically no levels of COVID in the ethanol, and that's why it's a control and what it's for. And then the results. Do a quick control save, drink some coffee. So, in the nitric oxide nasal spray, the absence of virus was found to be optimum. We'll have to learn what optimum means in their opinion. So, incubation of the nasal spray with Delta variants for 2 minutes led to a significant reduction in virus. So, it acts very quickly when it comes in contact with COVID, the nitric oxide nasal spray. And it has good cytopathic effects, so that just means it's killing the COVID. P-value is very significant, so that's good. Essentially, you want something less than a 0.05, so this is 0.0021, so that's very, very good p-value. CT value is found to be significant. Here we go, give you a better view. Significantly higher. In the case of treatment of Omicron variants with the nasal spray, the CT value is found to be significantly higher at 2 minutes and 15 minutes. Okay, so this is saying that when Omicron was treated with the nasal spray, this is another reason I don't like preprints, is because they have this stupid thing and it messes with your highlighting. So when it was treated with the nasal spray, the concentration, so a high CT value is correlated with, let's see if I can remember. I'm going to have to Google this. I always forget which way it goes. That's not my... I think it's a high CT value correlated with high COVID levels. So lower CT values are higher levels of viral RNA. So there we go. Okay, so so that's opposite what I was thinking. So it's the Omicron variants with the nasal spray were had it was reducing. Okay, cool. So it was being more effective over time. So the longer it was in contact with the nitric oxide was in contact with the COVID, the more it killed. And this was significantly lower after 15 minutes compared to treatment and control. But again, this is a static solution. In a human, you're going to be increasingly producing more and more mucus, because this would be going in your nose. So your mucous membranes in your noses are constantly flushing, and that would mean that any sprays would be, getting flushed out just through dripping out of your nose. Two, they could be being dissolved just by interaction with your nasal immune system. Or three, it could even be absorbed through your skin into your blood system. So those are different ways that you could have this be eliminated from your body over time. So you always have to be careful with these types of studies because you have to remember that they're taking it in isolation in a petri dish. You're incubating it, so all it's showing is that it is effective, but it's not telling you for how long it's effective. So that's always something you need to keep in mind with these types of things. And then I haven't looked through comments in a little bit, so let me just double check, make sure there's no things I missed. We're looking good. Okay. And then, okay, so the conclusion, and this is just the abstract. We haven't even gotten into the meat of the paper yet. So this is just the high-level thing. So this tells us what we should expect, and it gets us familiar with terms, what the general consensus of the paper is, and then we get to the fun part. Well, I think it's the fun part. It's when you learn how the authors think, how they did the study, you get to critique what they were doing, etc. So their conclusion is that the nasal spray exhibited significant antiviral activity against Delta and Omicron variants. So they're saying with their conclusion that it kills it. Nice. I agree with that from what they're saying so far. The results of our study support potential applications of nasal sprays as a post-exposure prophylaxis agent to reduce the severity of infection and control secondary transmission for multiple variants. So they're saying, again, they're not saying that these are going to be effective for preventing COVID in a person that is not infected. They're saying that it's controlling secondary transmission. So that means that a person with COVID, this could be used as a way to decrease their transmission levels through their nose. But again, you're not really going to be exhaling the majority of your aerosolized stuff through your nose. It would be through your mouth. That would be the issue. So it's like, if it was adapted for a throat spray, maybe that could be more effective. I don't know. But these are the types of things we want to think about when we're reading these. All right, so now we're into the introduction of the paper. So we got through the abstract. My cat's here. She purrs very loud. Take a little coffee sip. And like, this is the type of thing I'm just like trying to show you, like, yeah, you read through the paper, you're going to have questions, you're going to look things up, you're going to pause, you're going to come back. Things like that. It's not like, it's stuff that anyone can do. It's just being thorough and thinking deeply while you're reading. So, this is just a general introduction. For people that are familiar, none of this is going to be surprising. So it's going to be like COVID declared by the WHO in March 2020. 3 years later, we're at an inflection point. Levels of immunity to COVID is limiting its impact and reach. That I don't agree with because COVID decreases your immune system, so I make that red. So I don't agree that there's a high levels of immunity. However, recent surveys revealed that almost 90% of experts believe that COVID will continue to remain endemic in pockets of the global population for years to come. Also, that's a crap statement because if it's remaining endemic all over the globe, that's literally the definition of a pandemic, because pan is everywhere, endemic is it's unique to local populations. So that's just not a nice sentence. That's either they misunderstand it or they're being a little not genuine. And then nitric oxide nasal sprays, Febispray, has been approved for the COVID-19 treatment by the Central Drug Standard Control Organization. I never know when you should do an S versus a Z. I feel like that's a European versus American thing. Anywho, in India, as a part of the accelerated approval process, and several other countries have approved NONS, the nasal sprays, branded as a Vir-XTM, or Enovid, so that's what was mentioned earlier. And that's the one that made me find this paper, so I'm just going to highlight that. In this, because I do want to read more on this. I think I have a few more papers on this subject, but I haven't been able to find too much. As an antiviral agent for reducing COVID from the nasal cavity, several studies have demonstrated that a high nasopharyngeal viral load is associated with a higher transmission risk of COVID. Nitric oxide solution was shown to have a significant and rapid antiviral effect. In vitro studies conducted at Utah State University in the U.S. reported a 99% reduction. So I'm just going to quickly, that made me wonder, where's this study coming from? So this is coming from Bengaluru, India, British Columbia. Okay, so it looks like it's UK, India collaboration. So they're just citing a paper from Utah. Anywho, so in Utah, 90% reduction within 2 minutes. But again, is that holding up over time? That's the big question. Can cause significant reduction in viral load compared to normalized saline benzalkonium chloride placebo nasal spray. This showed a decrease in the secondary attack rate in the NONS group during 7-day treatment period compared to placebo. COVID-19, close contacts, proportion. So that's interesting, so we'll highlight that. We'll make it green because that's a fun little tidbit. So in another study that used this Virax nasal spray could reduce the spread of COVID between individuals in close contact by a statistically significant proportion in the infection rate. So it dropped from about 26% down to 6%. So this is a really nice thing saying that, hey, if you have COVID and you take this nasal spray and you, I'm assuming, take it at appropriate intervals, what those appropriate intervals are to keep your viral load in your nose down, that's TBD. But people that took this nasal spray prevented, so that's about 20% of household infections. So that's a neat little thing. Interesting. That's quite a lot of people. So overall, 11.4 patients reported some adverse events from the nasal sprays. We'll have to see if they list what those adverse events are. But that's quite a lot of people, so this probably wouldn't be great for a population level control. But there were no serious adverse events, suggesting that they may play an important role in preventing the spread of COVID since the outbreak of the pandemic. 96% of amino acid residues in human spike protein have undergone mutations, blah, 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 blah. Yes, of course, we know the virus is mutating. 2 such variants are Delta, first detected in India, Omicron variant, first detected in South Africa, blah, blah, blah. Just telling us about history of the variants. I already know all this. The virus undergoes frequent mutations, obviously. Various cell lines have been used extensively for characterization, so that's just a fun little thing to highlight, so that we're going to highlight yellow. So various cell lines have been used extensively for the characterization. So these are COVID cell lines, not like COVID viruses, but cells that are used in conjunction with COVID virus. And they're good because they're deficient in interferons and therefore support viral replication, which also makes them ideal for studying antiviral activity. So that's neat. And both ACE2 receptors and this TMPRSS2 are expressed in the cacao-2 cells. Cacao. It's probably CACO, but sometimes I just like pronouncing it. Cacao-2 cells. Let me just see if anyone's commented stuff. Plus, you can't have immunity when it continues to mutate at high rates. Exactly. Plus, you can't have immunity because it continues to mutate at high rates. Yeah, because there's easy, if it's spreadsheet Saturday, what do we call Sundays? Study Sundays? I don't know. I'm not good at naming things. Yeah, I just figured this is a good thing to go through. It makes me read papers more. Good morning.
What did you say?
I said good morning.
Is it?
I don't know. So antiviral effects have been described by Worcester. It's Sunday study. I like it. Sunday studies. So antiviral effects of not nasal sprays against COVID has been described previously by Winchester et al. And Miller. In this study, we aim to assess. Ow, donut, that hurts. We aim to assess the in vitro effects of nasal sprays against the Delta and Omicron variants of COVID. So then they go through the material section, how they did the study. So this is really good for people that want to try it and replicate studies. Hi, I'm just going over papers. I did. I was out of coffee.
Okay, but you did ask me because I woke up when you got up.
You told me not to bug you because you were sleeping in. Those are your exact words.
No, Patrick, you could have texted me, and so...
Okay, in the future I will. I apologize.
What is this hand for?
You can have the rest of this.
I don't... What is it?
It's a mocha.
Oh, my hands are inside out. Did you already drink that?
Yeah, I already drank that. I just needed coffee or else I wasn't going to be able to go grocery shopping later. So what the material method section does is it makes it really easy for people that want to try and replicate the study. That's actually a really big thing in science that is lacking is replication studies. Everyone gets funding and all the great stuff comes from coming up with new and novel ideas, but not a lot of people go and replicate it, and that's like something that we really need is for people to go and be like, all right, yes, I'm going to follow their methodology, see if I get the same results, and that's something that's really lacking. And a lot of times when people do that in the future, we find out that there was a flaw in like the data analysis, or they manipulated the data, or some et cetera, and that's prevents like so a good example is like the tangled tau proteins in Alzheimer's research. We've learned that that was a false flag, and that the methodologies were and the findings were a little incorrect, misleading, and it put us on this huge path and huge funding focus. So like we spent billions of dollars studying these tangled tau proteins in Alzheimer's, and it was not the right way to go, and it was all based off of a misleading study. So like that's the sort of stuff that can happen and what can be prevented by having timely replication studies on important findings. So, 1 second, I have to go get water, or else I'm just going to get progressively more hoarse. GP, you better not be pooping on my clothes. Allie?
What?
GP was just trying to poop on my clothes.
What do you mean he's trying to poop on your clothes? How do you know?
He has a face when he tries to poop on my clothes. I just barely caught him.
It's possible.
Look, you know he's walking his whole litter box of shit. That was good. Crisis averted. My cat was gonna poop on my clothes, and I would have missed it. Okay, so let's see. Yes, I've heard that before. More replication studies. Yes, we need lots of more replication studies. So that's sort of what these are for. So if you're not interested in that type of thing, then you can usually skip the methodology section. So I'm just going to glance through just because I like to see if there's anything that like pops out. So like this is just like a general overview. They're saying that we're going to use these cells to look at it, and they already explained why they're using the cells. So that's interesting. They're using various exposure concentrations of the nasal sprays. So that's going to tell us, there's probably going to be a chart later on that's saying, hey, this is the effectiveness of doing a 0.5 nasal spray up to a 30% nitric oxide nasal spray. They're doing some well plating for this stuff. They've got absorbance measurements, 450 nanometers. Tubercles modified eagle medium, love that name. It's just a growth medium that helps to culture cells so that they don't die. Virus to nods mixture was 1 to 10, so for every 10 milliliters of liquid solution, there's 1 milliliter of virus. They're doing some cell viability, so this is going to be fun pictures later, where we're going to see likely a green versus red, so a live versus dead cell type thing. That's where the absorbance is coming in. I was like, why are they doing an absorbance testing? And then it's because of the fact that they're doing cell viability. I'm just going to double check that didn't miss any comments. We're good. CPE. What is CPE? Control F, CPE. Cytopathic effect. That's weird that they decided to abbreviate that. Can you guys still see that? Yeah, we're good. Okay. Let me move this over a little. Yeah, so we're in assessment of cytopathic effects. That's interesting. I didn't know that. SARS-CoV-2 is known to show plaque-like cytopathic effects. So that's probably going to be something I want to study later on and look deeper into. Because if it's causing cell fusion, cell death, apoptosis, destruction of the epithelium integrity at a general cell culture level, that would be interesting. Because we know like the brain cell fusion stuff that's going on. I hadn't seen anything about apoptosis before. And we know destruction of the epithelium, the inner lining of the cell walls. So it'll be interesting to see. That would be something I'd be interested to follow up with. And then assessment of viral load. And then a lot of times, just for like reference, we have like in science, like you don't have to do everything from scratch. I guess I'll do a new color typing. So RNA isolation was performed using the NucleoDx-RT RNA extraction kit. You can literally buy kits that are for specific purposes, and this, you just literally follow the instructions of the kit, and then you can do fancy stuff. So like, you don't have to invent the wheel from scratch every time you do science, because like, you're not going to have everything accessible to you all the times, but like, all the things that you need to do, a lot of the times you can purchase. Again, that is a financial barrier a lot of times, but like, this NucleoDx-RT RNA extraction kit exists. You can purchase that, and it makes this type of study a lot easier. And it looks like they use too small of sample size, or like sampling, so that's going to be a bad note against their study. So the quantification of RNA using fluorometric or photometric methods was not possible due to the low concentrations of viral nucleic acid. Viral nucleic acid, yeah, that is the right way to say that. So that's just saying that there was not large enough volumes of RNA in their samples to be able to visualize it appropriately, so maybe we won't. That's disappointing that they... We'll have to see further down, but that's essentially implying that the study that they did, they weren't able to look at things using fluoro, even though they're doing fluoro. All right, now we get into the fun. So there's, sorry, I skipped over an important section, statistics. So this just tells how they're doing the stuff. So they're doing students t-testing for comparison of means. So that's a fine way to do it. We'll look at their test question further down, but their p-value, they're doing a 95% confidence, 0.05 alpha value or p-value. And then they're saying that they use GraphPad Prism as the program for statistical analysis. And now we're in the results. This is the fun part. So there's a gradual decrease in cell viability. So that's just saying that as the concentration of nitric acid increases within the nasal spray, more cells died. And that apparently a 10% nitric oxide spray was ideal or optimum. So I'm guessing, this is a guess, I don't know, is that the at higher concentrations of nitric oxide spray, you would actually be damaging some of your nasal cells, like the good cells, quote unquote. So this is like the balance concentration, where it would be enough to kill the COVID that's being excreted from the mucus, but not enough to damage the nasal cells to a significant degree. That's my guess. We'll probably learn later in the conclusions or discussion. So this is a little plot that's looking at cell viability. Yep, I think I was right. So this is looking at the Vero cells. So these would be like the human analogous cells. So you want a high level of cellular viability, meaning that these are the percent of cells that are alive. So once you start to get to 20% concentration of nitric oxide and 30% concentration of nitric oxide, these cells that are your quote-unquote human cells are dying off. Like at 20%, you're killing over 50% of the cells that it's coming into contact with, the quote-unquote human cells. So that's why you would not want to go above a 10% concentration of the nitric oxide, so that you're not damaging your nasal cells. Delta strain was pre-exposed. Wait, what? Yeah, we already know that. They said that earlier. They showed that the nons could effectively neutralize the strain for the Delta variant. The neutralization effect was comparable to that of the positive control. So that's saying that it would killed like pretty much all the COVID at 70% in 70% ethanol. This is consistent with the virus. Non's mixture was diluted. And they did 3 different test periods of time. So this is the duration that they were mentioning earlier that I'm not really sure what they're talking about. So they tested after 20-second exposure to the nitric oxide, 2-minute exposure to the nitric oxide, 15 minutes exposure to the nitric oxide, and tested at those different time intervals. And then let me just see if I missed any comments. We're good. Okay, so moving on from there. So now we get into fun graphics. So these are microscopic images showing the Vero cells. These would be the quote-unquote human cells. So this is, let me zoom in. So right here, these are just cells. These are cells in the media. These are cells in PBS. These are cells in ethanol. And then, I have to scroll over, I think, a little bit so you can see it. And then these are the cells in the nitric oxide. After the top one is without, what's HEPS? HEPS. HEPES. We're going to have to Google that one. Or not Google, Control F. HEPES. Oh, okay, so HEPES is hydroxyethyl 1 piperazine thane sulfonic acid. Not sure what that is. But I guess their point is that it doesn't matter if you have this chemical in, because what I'm seeing is that they're pretty comparable between the two, with and without it. But what is the interesting thing here from the pictures is that, like, you have lots of cells in the media, lots of cells in the PBS, you have basically no cells with the ethanol and basically no cells with the nitric oxide. Showing the reduction in Vero cells in the human-like cells treated with these treatments. Then we'll look at the graphs, the figures. So this is cell viability of Vero cells with and without HEPES upon infection with the nitric oxide spray for the Delta variant. Yeah, it's basically just showing the same exact thing. It's a redundant figure. Same thing with this one. Cell viability, Delta variant was comparable to that with ethanol treatment. So now we're at the part where we already know all the findings. It's just seeing if there's anything interesting that's popping out in the plots that they didn't discuss. Is the point we're at in the reading. Oh, here we go. This is fun. So this is the 20-second exposure, the 2-minute exposure, and the 15-minute exposure. So this is the cell level. We want to see that this cell level is staying pretty high. Oh, that's weird. It's probably just an artifact. So the black here is the nons, the nitric oxide spray. So we have this one, and this one. And at 22 and 15, it decreases from the normal cells. It's about the same at 2 minutes, and then it decreases again at 15 minutes. But so this is the error bar. It's weird that they're not doing a 2-sided error bar. They're only doing a 1-sided error bar here, but it's just usually the same exact level as the one on the top. No, these are significantly different. So yeah, it does lower the amount of live cells. It does damage some of your normal cells by using this spray. And then we're on to the discussion where the authors give their final thoughts on everything that we've learned. So this is an interesting point. So the research has shown that the virus can traverse the mucosal layer of the airway using modal cilia as tracks to access the cell body. That's really cool. It attaches to the airway microvilli, 24 hours post-infection and triggers the formation of highly branched microvilli. These microvilli help the virus move back into the mucus layer, and so I'm just going to highlight all of that. Help the virus move back to the mucus layer and disperse throughout airway tissue using mechanism of mucociliary transport. So essentially what that's saying is that the spike protein is sort of like rolling along these microvilli, and it's being transported into the airway tissue, sort of following along this little tract. And again, that's an interesting thing that I want to follow up with, is where they're saying that it's fusing the cell membrane and climbing in, or I need to probably read this study that it's referencing. So that's another thing about these papers, is that when you see an interesting fact like this, you can find the source of the paper. So it's source 32, and I can scroll down into the references. Source 32 would be this paper right here, called Replication in Airway Epithelia Requires Modal Cilia in Microvillar microvillar reprogramming. So that's how you can find more articles when something's interesting. So post-infection, there's a 24 or to 48 hour period during which the virus is contained in the nasal cavity. That's also a nice fact. So this would be after your quote-unquote infection or infectiousness is over. There's still you're still producing COVID in your nasal cavity for about 2 days afterwards. So that's a good thing to know. And this seems to be postulating or theorizing that how the nitric oxide spray works is that it suppresses furin, and which is a protease, so an enzyme for proteins that requires COVID, that is needed for COVID to replicate. So it stops that enzyme from being released or produced, and that prevents the COVID from being able to replicate. And this happens by reducing calcium levels in the cytosol. So it's probably blocking the calcium channels. So this blocks calcium from entering the plasma that's inside the cells. Probably because the nitric oxide is creating a negative charge on the outside of the cell. And when you have passive diffusion, when you have high negative charge outside the cell, the positively charged calcium ions inside the cell will be forced through those passive diffusion channels to the outside of the cell, which reduces the calcium levels in the cell by pulling it out of the cell, if that makes sense. And then every good study has a limitation section. Can you test influenza in wastewater? Yes. You can do genetic testing, genomic testing on it, just like we do for COVID, and that tells you what the specific strain of the influenza is. You can also just detect its presence there as well. We can do that for any virus. And then limitations of the study. Yeah, they did weak limitations because they didn't focus on the big issue, which is, okay, we're showing that it has an effect in a static medium, but our bodies are not static, they're dynamic, so we're constantly producing mucus. How long will it stay in our nasal sprays? How long would we have to refresh the spray to maintain the same effect? Why what? Why can we test influenza in wastewater? Or why in terms of what I just said? See above comments. Can you test influenza in wastewater? Why can we? Because viruses have RNA. RNA is the genetic code for the viruses themselves. So, for example, if we go to wastewater scan, yeah, I'm not sure if your comment came through. It just shows me, can you test influenza in wastewater, and then it says why. But in the wastewater, it has the viral RNA, and it has any viral RNA that we shed through our feces and our urine. So things that they test for in wastewater scan is things like COVID, the flu, RSV, human metapneumonia virus, EVD68, and parainfluenza. These are the types of things that we can test in the wastewater. And how they do it is they take wastewater samples prior to when we do the processing to clean the wastewater. They basically just stick a test tube in, pull it out, send it to a lab. You sort of separate the you just pull samples from the waste, and then you can test. You basically like run it through a machine, and you can detect what types of RNA signatures are in there, and was pulled in for COVID. But she doesn't look at wastewater. Sorry. She doesn't look at wastewater, and I'm just curious why she doesn't and was pulled in. Who's she? She doesn't look at wastewater, and I'm just curious why she doesn't and was pulled in for COVID. So I don't know who you're referring to, because for some reason the comment's not coming through on the person's name. But I'm not sure why they would not look at wastewater. Wastewater is a very early indicator for when COVID is. She does flu VE modeling. Oh, cool. So flu modeling, maybe the person is just really interested in the flu. The flu has been around. Dr. S. Kobe. Okay. So let me just. We have Google, so we can. Dr. S. Kobe. So this is Dr. S. Kobe, an ecology and evolution of pathogens person. Their focus is on dynamics of viruses and immune responses. It seems like their research is focused specifically on the flu. So that's probably why they're just modeling the flu. But their modeling for the flu could likely be maybe shifted over and also used for COVID, but that's probably why that person is focused on just the flu. And also, we have a long history with the flu. It's been around for a very, very long time, unlike COVID, which popped up 4 years ago. So we have large pools of data, and we've been tracking it very well for a long period of time. And it comes very seasonally, very regularly. So, like, this is California's flu, for example, for from 2022 to 2023. And like you have a little bit of noise down here, but you have a very clean, clear signal in the winter from about October 16th to February 2023. And if we actually enhance this to a national level, it'll probably make it even clearer. So it's going to take a second to pop up all of the different states. Here, let me go. So she's on, I support her because she got pulled in for COVID. She's on the team who picks which flu vax we get each year based on seasonality modeling. That's really cool. That's, so she's definitely an expert in the flu. Hi, Bex and Bean. How are you doing? Nice to see you again. So right now we're just discussing the flu. So this is the wastewater scan dashboard from 2022 to 2023. They have very clear signal nationally for the flu. So really it just happens once a year. And then this is October. So again, this is the same period. It looks like it's different. So let me just chop it down to a single year view. If I can. Page is unresponsive. I'll wait. So I might have to not be able to do that. But the flu just has a singular signal in a year. It doesn't really happen the rest of the year. It just happens from essentially October to February. So it's really easy to model. It follows a regular pattern. COVID is still evolving, so I think wastewater scan didn't like that I'm modeling every single thing. I'm essentially pushing their program too far. She's so nice too, insanely smart. Thanks for explaining the difference for my non-scientist brain. Yeah, no problem at all. That's science communication. So that's that to answer your initial question. That's why she doesn't focus on COVID. But let's pop back to wastewaters. No, that's the wrong one. Sometimes I go too quick. Wastewater scan. So this is just California. And so you can see this is data from 2021 to the present. And like we had, I'm gonna not use my mouse because it does all that glitching. We had a little bump. So I always think of it like heartbeats. So we had like a very spread out first year. The second year, higher signals, closer together. Third year, pretty much all year. So it doesn't have the same type of irregularity. So it's a lot harder to model, and there's not tons of data because with the flu we can go back like however many, like 80, 100 years of manual tracking and our computer tracking versus only 4 years of data. But going back to this paper that we were looking at, so our study, we're looking at nitric oxide. We've been reading through this. This is a preprint, so the information will probably change by the time it gets to a full review, but I just wanted to get some basic information on these sprays because people have been asking me about them. And they need some water. They say that in their conclusion, short and sweet, that they confirm antiviral efficacy of the nasal sprays in Delta and Omicron variants. It can work very quickly, as quick as 2 minutes of contact with the nasal spray. And it can be used as inhibition of viral replication and early neutralization of the virus. Again, this is in a person that is producing COVID, not a study that would say anything about a person that is trying to prevent COVID. And the biggest limitation that is not addressed in this, again, is the fact that this is a static system as opposed to a dynamic one. So then we can just go through and do the highlights of this paper. I so appreciate your content. I've learned so much from you. Thank you. Yeah, no problem. That's sort of what I'm doing, is because there's so much, or what I'm trying to do is because there's so much misinformation out there, and people are just like, I'm an expert, I know what I'm talking about, when they haven't like been reading the literature and staying up with stuff. And I'm like, okay, well, let's not focus on qualifications. Let's focus on what the literature is telling us, what we're seeing, and what the data says. And let's just share it with people, because that's what we should do, and show people how to think about science and what it looks like. And like, it's literally just like a mode of thought. And it, like, as I walk through these, and I'm going to try to do this a little more regularly on lives and stuff to like read through papers, people should be like, oh, that's not too bad, I can do that. And like, I do have like tons of science background experience from a master's degree, a PhD program that turned into a master's because of issues beyond my control, and a college degree, and essentially 11 years of advanced science education plus whatever I'm doing now. So like, I do have that additional stuff, so I can, like, as you saw me walk through this, I can be like, oh yeah, so this is what I'm familiar with, and this is why it's weird, or this is why blah blah blah. But anyway, so let's summarize the paper real quick. So we're looking at nitric oxide sprays. We found that a 10% solution is good. The reason that they don't want to go higher than a 10% solution of these nitric oxide sprays is because it will actually damage your good cells, your nasal cells, at higher concentrations from that, so that would not be good. But at 10%, you are not damaging a significant number of nasal cells, but you are also killing COVID. They looked at Delta and Omicron BA1 in their experiments. They tested at different time periods at 20 seconds, 2 minutes, and 15 minutes, and they found that it increasingly killed COVID over those increasing time periods, and that at 2 minutes it killed a significant proportion of COVID. So this could be very good for people that have active infections. We saw that, so again, this is focused on secondary transmission control, not preventing a primary transmission. So these are for people that already had COVID. The really interesting thing here is that when they prevented household infections, there was a 20% reduction in household infections using the spray. It dropped from 26% down to 6%. However, there's a high instance of adverse events. They didn't discuss what these adverse events were, but there's 11% of people that use the spray have an adverse reaction to it. So that's something that is probably a high contraindication against this, something that would make it something you wouldn't want to do. We learned about these human line cells. I'm not sure if they're human line cells, but they're cells that they're treating as human analogous that have specific qualities that make them really good for studying viruses. We learned about the methods that were used. Unfortunately, they didn't use large enough samples to do fluoro, which is really sad because they ran the experiment and they talked about the experiment, but they didn't get any results. And then basically what they were showing in here is all very, very redundant. They're just filling up space. It's just showing what we already talked about. And then they talked about this really interesting thing about how the microvilli in our nose can sort of help transport COVID, so COVID can stick to these and roll. I'm almost done. And then fusing there were statements in here about how it can fuse cells. So I have only learned about the fusion of neurons. I'm interested what other cells COVID can fuse. So I'm going to follow up with this citation 32. And that another interesting fact we learned is that when you are no longer infectious, quote unquote, Once the virus, you're in a post-infection period in your COVID infection, there is about a 48-hour window after that where you're still producing virus in your nasal cavity, so that could still be a transmission source to other people 2 days after you're quote-unquote cleared the virus. We learned about a little bit of cell channels that we talked about. And yeah, that was the paper. Cool. So that's the paper I'm going to read for now. I'm going to probably go grocery shopping now. So thanks for stopping by.